Hello again, biologist. Mr. Kinnear here with another video lesson for higher biology. We're on Unit 1, DNA and the Genome, and this is the second of our video lessons, and we're looking at replication of DNA, key area 1.2. So this is where today's video lesson fits into the unit. We've already looked at the structure and organisation of DNA, so replication of DNA next builds upon our knowledge and understanding of the structure and organisation. So here's what we're going to be learning today. When does DNA replication happen and why? What is required for DNA replication to happen, so the molecules that are important for it to go ahead, and how does it happen? So what are the steps and stages in DNA replication? We touched upon DNA replication within National 5 Biology last year in the process of mitosis. So we know that cells are replicated, so a parent cell becomes two daughter cells. And why is DNA replication important? Because that is the process that creates an identical copy of its DNA. So this happens before the cell splits, making sure that each daughter cell will inherit an identical copy of the DNA. So each and every single one of your cells will have a full complement of your chromosomes after each cell division. So now let's look at what is required for DNA replication to occur. We need to start with a DNA template. If you're going to the photocopier to make a copy, you need to stick in a bit of paper, and that's what we're doing with a DNA template. This is the bit of DNA that we are going to copy. We'll require primers, we'll require nucleotides, and as we discussed in the first video, there's 4 different types of nucleotides. The phosphate's the same, the deoxyribose sugar is the same, but it's the base at the end, means that there's 4 different nucleotides that could be available. And we require enzymes, so 4 different enzymes we're going to talk through as we go through the different stages. And last of all, a supply of ATP. We are going to be building a polymer, we're going to be building chemical bonds between nucleotides, so we need energy to create those chemical bonds. DNA replication is often referred to as a semi-conservative process, and hopefully from this image and the 3 steps you can maybe see why. What we start off with is one ladder, like we described in the first video, of red, and actually what we end up with at the end is half a ladder, which is red, half a ladder that is blue, and 2 copies of them. As you can see, when we add in our new bases, each of our 2 new strands is half old and half new. So that semi-conservative process means that every time we replicate DNA, half of each copy is old and half of each copy is new. So we're conserving 1/2 of the DNA each time we replicate it.
So over the next few slides, I'm going to talk you through the individual steps involved in DNA replication. And I'll introduce you to the enzymes that are involved and what they do in the process. So here we have what looks like a ladder on its side. So effectively a small segment of DNA. We've got 3', 5' at the top, 5', 3' at the bottom. So that's an anti-parallel strand, as you may remember from the first videos. So this small segment is going to be replicated. Obviously, DNA replication happens across all your chromosomes, all your DNA at the same time. But we're going to look at the process at this small segment. So we've got hydrogen bonds there in the middle between our bases. And as we know, it's a semi-conservative process. So the first thing we need to do is split this double helix or double strand into 2 individual strands. And that is done by an enzyme called DNA helicase. So simply, helicase, its job is to split that helix into 2. So it breaks the hydrogen bond, separates the strands. There it goes. Okay, so we've now got 2 DNA templates. Started off with a double strand, we've now got 2 single strand templates to work on. So now we have our 2 individual complementary strands ready for replication. Primase, our new enzyme, is going to come along and synthesize some RNA primers, which are small sequences of complementary RNA. So like primer paint, it's a coat of paint that allows the second coat to stick better. Our primers are going to allow our second enzyme to come along and stick. So primase has come along, it's added on those primers. So as we can see, they're complementary, but what it does do is it has uracil here instead of thymine. So it's RNA rather than DNA. But these primers allow our next enzyme to come along and know where to start. So the next enzyme we have involved in this process is DNA polymerase. As its name suggests, its job is to create a DNA polymer, so a chain of nucleotides. And it does that by adding in free complementary nucleotides to the parent strand, starting at the primers. Okay, so DNA polymerase comes along, binds to primers, that's where its starting point is, then it moves along the chain. And as you can see, wherever we have guanine on the parent strand, we add in cytosine. Wherever we have cytosine, then we add guanine, and so on and so forth. So we're adding in complementary strands as it moves along. However, there's something a bit strange. This strand along the top is being synthesized in a continuous manner. This strand along the bottom is being synthesized in fragments. So why might that be? It does relate to the direction in which DNA helicase is moving. So it moved this way. It split our double helix in two. So this is where it's being unzipped effectively. And you'll note that DNA polymerase was moving in this direction on this chain and this direction on this chain. And that relates back to our 5', 3'. We discussed this in one of our first videos. The chain of nucleotides is extended or grown by adding nucleotides to the 3' end. So as we can see here, we're moving or growing or replicating in this direction. Okay. However, DNA strands are anti-parallel. So it's upside down effectively. So in this chain, we're actually going to move this way when we're adding nucleotides in. So the enzyme DNA polymerase can only add nucleotides to the 3' end of a new DNA strand. So it moves along the old strand or parent strand or template strand 3' to 5'. However, the replication fork effectively is opening at different ends on our different strands. So our top strand would be referred to as the leading strand. It's being replicated continuously because where DNA polymerase needs to add a new nucleotide is continually being freed up by DNA helicase unzipping our original template strands. The lagging strand, our bottom strand on this diagram, has to be replicated in small sections because it's going in the opposite direction of DNA helicase and the replication fork being created. These small sections of DNA formed along the lagging strand are referred to as Okazaki fragments. They were discovered by husband and wife team, the Okazakis, and therefore have been named after them. So our leading strand up here is being replicated continuously because DNA polymerase is adding in new bits of DNA, free nucleotides, in the same direction that DNA helicase is moving and splitting our double strand. However, on this strand, our DNA polymerase is moving this direction, which means this part of the DNA was only available after this part and after this part. So that is why we get these small fragments, because DNA polymerase can only move in this direction along the lagging strand. So we're almost got two identical bits of DNA here, but what we do have is still some uracil base in there. We've got some RNA primer still in there. So DNA polymerase comes back and replaces the RNA primers with DNA. So we're almost there. However, we do still have gaps on our lagging strand. So we've got our last enzyme ligase, effectively the glue. What it's going to do is stick or seal these fragments together and produce two new identical double helix. So this is a fantastic gift from Amoeba Sisters. They do put together some amazing videos and GIFs and other sorts of science communication. And you can clearly see 3 of our 4 enzymes working here, replicating DNA, helicase splitting the helix, DNA polymerase in green, creating our new strands, primase adding in primers for DNA polymerase to join onto. The only thing it doesn't show is ligase connecting the Okazaki fragments together at the end.
So in order to follow up this video, it's worthwhile making your own notes on the diagram so that you have your own version of this. Obviously you can use Scholar and Bite Size to help you with that information. Key things you should be having on there are the enzymes involved, the directionality of replication on both template strands, and also the complementary nucleotides added at the 3' end of the new DNA strand, and the fragments of DNA joined together by ligase.
But have a look at this. So this is something new, just to blow your mind a bit. What do you think is going on here? So effectively, we've got what's known as replication bubbles. So what I've seen before, you normally get a replication fork, but what we're actually saying is that DNA replication happens in both ways on long strands of DNA, big chromosomes. So we end up with helices moving in both directions, splitting the DNA in both directions, and therefore creating these replication bubbles where our DNA polymerase is adding nucleotides both sides and multiple different regions of our DNA. That helps it happen faster.
The DNA replication that we've looked at so far is in vivo, as in a living organism. We're now going to look at the in vitro version, which is obviously within a lab environment. So polymerase chain reaction is a lab process which is able to replicate DNA, so we get multiple copies of that DNA template, which is very useful for a variety of different reasons, which we'll discuss as we go through. So the key concepts we're going to look at: what is PCR, how does it manage to amplify DNA, and the applications of PCR within research and medicine. So some of the things that we've looked at in terms of DNA replication in vivo are required for PCR, and some of the things are not required because it's a slightly different process, eliminating some of the in vivo requirements for DNA replication.
So polymerase chain reaction, you should hopefully understand the word polymerase. That's the enzyme we saw in our last video lesson that was involved in the replication of DNA. And a chain reaction, it's something that builds upon itself. So something that will increase in volume or increase in terms of reactivity as time goes on. It's also commonly known as PCR, and it's used to create many copies of a piece of DNA in the lab. So we're talking about it in vitro, so something that happens in a lab. And those pictures on the right hand side there are thermocyclers or PCR machines. So that's something that we could create huge amounts of copies of DNA in a laboratory. It'll sit there and work away for 2 or 3 hours, and incredible volumes of DNA could be produced. That's called amplification. So just like you would have an amplifier or an amp connected to an electric guitar in music, that enhances the volume, increases the volume, amplifies the volume. And that's exactly what we're going to be doing today, is amplifying the volume of a piece of DNA. And it uses complementary primers. Primers are about 15 to 30 bases long, and nowadays they can be made in the laboratory. So having spoken to somebody from the University of Aberdeen last year, you can actually go onto a website and simply type in the bases that you require for your primers. And what that does is help target bits of DNA, segments of bits of DNA that people want to amplify. And I think within 2 to 3 days, then you'll receive in the post a box with some vials of complementary primers that you'd use in your PCR machine. So just to give you a bit of background into the development of this process, the enzymes and reagents that are used in PCR are the same as those in DNA replication that we looked at in the last video. So DNA polymerase, primers, nucleotides, DNA templates, those are all loaded into a PCR machine. You can see that block being added into a PCR machine ready to amplify a sample of DNA. So everything that is required for DNA replication is the same as PCR. So it's loaded into those sample wells for the PCR process. It began in 1976 when Taq polymerase was isolated. That's a specific DNA polymerase, and one that we're going to look at in a bit more detail shortly. In 1988, it's commercialized and started used widely. In 1989, Science magazine dubbed Taq polymerase their molecule of the year. And in the 1990s, high quality enzymes were isolated that were very reliable. And again, the thermocyclers, those machines that we've seen in the past pictures, those are developed and therefore really revolutionizing the process and allowing for this to be used in a wide variety of applications. So what are the applications of PCR? Why is it so important? First one up is the Human Genome Project. That's something we will look at in coming lessons, but effectively it's mapping out the genetic information or genetic code for human beings. It helps us understand a bit more about genetic disorders and diseases, etc., and helps us tackle them. DNA fingerprinting, so forensics. If you've ever watched any kind of drama or documentary about forensics, you'll know that they can pick up very small bits of DNA. So from one single hair or a very small amount of skin cells. That DNA that's in that sample then is amplified using PCR, which then means they have lots more of the sample to be able to compare it versus the databases or the suspects that they have for the crime. And one that's really prevalent at the moment, detection of bacteria or viruses. So detecting the presence of the coronavirus is something that PCR is used for right now. So samples that have been taken from potential people that have the virus, they are then amplified using PCR. So then they can identify the strands of DNA that have come from the virus. Also, as well, it's involved in the production of potential vaccines and also as well tracking where the diseases come from. So having read an article about the spread of coronavirus into America, obviously they suspect it was coming from China, so they shut the borders to China, but actually the most amount of spread of COVID-19 has come from Europe, and they can detect that by using PCR to amplify the genetic strain and then track where the mutations have happened. And they have been able to identify the majority of American cases have actually come from a spread from Europe. So I'll share with you a couple of different news articles that involve PCR in this current situation. And last one is diagnosis of genetic disorders. So sampling people to see if they have a potential genetic disorder that may have an impact on them in later life. So that's one other use of PCR. So these are things that you will need to be able to say. An example of a question that could come in an exam is name an application of PCR. So there's 4 of them right there for you. So I've mentioned Taq polymerase previously in this video. It's a heat-tolerant polymerase, and as we go through the process of PCR, what you will find out is that it does use temperature in the process. Your DNA polymerase that you have to replicate your DNA during mitosis, its optimum temperature is 37 degrees. So it just wouldn't function in the process of PCR. So what they have is isolated it from a bacteria. You don't need to know the name of that bacteria, but they do live in hot springs. Therefore, their enzymes are adapted to work at high temperatures. So without this Taq polymerase molecule that it's adapted to work at high temperatures, the PCR process would not work. There are 3 stages to every PCR cycle. So melting or denaturing, annealing and extension. So you will need to know these 3 stages and what happens within those 3 stages. And that is one cycle, and the cycle is then repeated several times to amplify the DNA, and we'll show you just exactly how quickly and how much DNA you can get through just a few cycles of PCR. So stage 1 is melting or denaturing. And what we're going to do is we're going to heat up our sample to 95 degrees Celsius very, very quickly. And that obviously means that some of the enzymes that you would use normally in your DNA replication just would not work in this process. So what we're doing is we're going to break the hydrogen bonds between bases and effectively melts the DNA, splits it into. So we'll start with a DNA double helix, and then we break it down, and then we end up with 2 separate strands. So we're avoiding the use of helicase in this process, we're just using heat. So stage 2 of the process is known as annealing. That is when we cool the samples down to around about 60 degrees and primers bind to specific DNA sequences. And I'm going to be very specific about this word bind. People have lost marks previously by using the word add at that stage. Now if you say primers are added, that makes it sound like you open the machine up and that's when you pour the primers into the machine. And that's not the case. Remember that everything that is required for this process is loaded into your samples right at the start. So when it's cooled down to 60 degrees, That allows primers to bind to specific DNA sequences. And remember, those primers can be engineered specifically for the process of amplifying a specific bit of DNA. So stage 3 is known as extension. We're going to heat this up again to 72 degrees. That's the optimum temperature for Taq polymerase, so it's going to work really quickly. And it's called extension because we are going to extend our complementary strands of DNA, starting at the primer, as it happens in DNA replication. So the Taq polymerase will add on new free nucleotides onto the 3' end of each of our new strands of DNA. So that means after one cycle of PCR, we now have 2 double-stranded DNA sequences. And again, it's semi-conservative like DNA replication, so it's 50% of original template of DNA and 50% newly synthesized DNA. So it means after one cycle, we have doubled our DNA. And then that cycle is then repeated a number of times. So a quick numeracy check. So if our DNA is doubled each and every single cycle, start thinking about the number of copies that will be present after one target sequence of DNA has passed through 7 cycles of PCR. So you want to pause and quickly do your sums either in your head or on a bit of paper, and I'll tell you the answer. So 3, 2, 1. It is 128 copies. And let me show you how that happens. So one thing that might throw people on that last question is that you've got to remember that our template DNA will be double-stranded. So we actually have 2 copies there. So after one cycle, we have 4 copies. And crazy, after just 4 cycles, we've got 32 copies. And after 35 cycles, we could end up having 68 billion copies. So as you can see from that, we have exponential amplification. And now a cycle could be as little as 7 to 10 minutes. So think about how quickly you can amplify DNA in these PCR machines to get a huge amount of DNA copied in such a very short amount of time. So this type of graph may be something you might see in your final exams. This effectively is one cycle. So starting here and finishing here. Something you may be asked is how long each cycle takes. Now, obviously down here, we don't have an actual time, so we can't tell that from this graph. But what you should be able to identify from this graph is the 3 different parts of one cycle. So 3 different stages. So we're ramping up there to denaturation. So this time is denaturation. It then cools down to, in this instance, 54 degrees. That's our stage of annealing. And this stage is extension. Now, if you have a longer bit of DNA that you are trying to replicate in the PCR process, then this extension time may be longer because obviously we're having to add more nucleotides in because we're creating a longer chain of DNA. But effectively, you should be able to identify start and end of one cycle. This obviously denaturation is then beginning a new cycle, so separating out our new bits of DNA into templates for the next stage of annealing and extension.
So that completes our video lesson for Key Area 1.2, DNA replication. We've looked at DNA replication as a semi-conservative process producing 2 identical strands. It requires a template, primers, enzymes and energy in the form of ATP. And we've also looked at the PCR process, which is the in vitro amplification of DNA within a lab within a very short amount of time. So we've looked at both in vivo and in vitro DNA replication today. Hopefully that's been helpful. So as always, if you have any problems, questions or queries, let me know and I'll speak to you again soon. Bye.